under high heat conditions. Generally, we boil a solution to do this. Or we can throw in a little bit of sodium hydroxide, and that will render double-stranded DNA in a test tube single-stranded. Then we can reverse that insult. We can either go down to some reasonably cooler temperature, or we can neutralize the solution, and the strands will spontaneously, just through the help of diffusion, find their correct anti-parallel complementary partners and restore the double-stranded DNA. Now, there are conditions that matter. If the DNA is complex and dilute, this could take weeks or months. If the DNA is concentrated, short, and simple, it could happen in seconds or less. And so we have a little bit of control over how fast this goes on. But I can't think of any genetic technology that doesn't take advantage of this spontaneous property. So it's one that we'll see a number of times. Making things melt like this, sometimes melt is the word that's used. Denature is another thing to be said about what's happening here. When these strands come back together, that can be reannealing. Hybridizing is another word that's used for that. I guess denaturation is the word they're using here. When we take a single-stranded molecule, generally in a laboratory situation, and we label it with something that we can find, a molecule that glows in the dark or emits radiation or something like that. Sometimes we attach a great big enzyme so that it can conduct a color change reaction wherever it's located. That probe can be used to find its complementary anti-parallel target somewhere else. Kind of like that. You just have to remember, if you're asked to perform this sort of a function, that it finds anti-parallel complements, and students generally don't include the anti-parallel aspect of that relationship. So I've seen a number of college level questions designed to see if you're thinking about the anti-parallel backbones. The simplest, most straightforward experiments that can be done with a probe are to probe chromosomes in situ, chromosomes as they are. Take the cell away, get rid of the cell membrane, get rid of the nuclear envelope, get rid of the cytoplasm, and generally these chromosomes, as they are, just DNA wound around histone proteins. They can stick to a glass slide. You could dip the glass slide in sodium hydroxide and then probe that with radioactive probe or with a glow-in-the-dark probe. And you can see where sequences are located on the physical chromosome, which is often a very convenient thing to do, especially like they did here with 6 different probes in one experiment. You can certainly tell what the order of the genes is. I can think of other ways to find the order of genes on a chromosome, but it's an awful lot more work and takes a lot more time than this is so visual and so direct. So that may be a technique that you see that simply takes advantage of the hybridization of a probe to chromosomes in situ. A little bit older fashioned, but still modern techniques that have replaced the southern blot work by the same logic, and so it's good to not forget the logic of the southern blot, even if in reality people don't tend to run these much anymore. It does tie together things that we've talked about. DNA restriction, cutting a complex genome into small bite-sized pieces and then spreading them out using electrophoresis into all the different lengths. Thinking about those million different fragment sizes, moving the DNA onto a solid support, and then probing with a single-stranded labeled probe to see where a particular sequence might be. Not on a chromosome now, because the chromosomes have been rendered a DNA chemical. So this is no longer in situ, this is in a southern blot. But you can see signal, you can at least find the restriction fragment length. So sometimes this is called a restriction fragment length polymorphism. So sometimes called an RFLP. The lawyers still may be using RIFLIPS. They got quite famous during the O.J. Simpson trial. When was that? In the 90s, so it's going back a fairly long way. We saw an example of this kind of technology here. The extent of the probe is given. The small map around the probe sequence is given. The positions of endonuclease target sites. One of them is marked with a star. That one must be disrupted by a particular point mutation. A depiction of a gel, size standards shown. Certainly you guys are expected to know where to put the larger and where to put the smaller size standards. And then using this map, can you predict where homozygous, heterozygous, and mutant homozygous signal might land on a gel like this? Even if a southern blot is old, this is the same way that more modern technologies look in the environment of some kind of a gel electrophoresis. So that certainly does make sense. We'll see some more modern techniques today that kind of have replaced, in terms of speed and expense, have surpassed southern blot. This is one of those approaches using a very specific probe. The difference between probe 1 and probe 2 is just that one base. And the trick here is the hybridization conditions. The concentration of the salt, the precise temperature is worked out so precisely that the difference between those two hydrogen bonds determines whether that probe sticks or doesn't stick. And again, a lot of the modern technology involving big machines that all the hospitals now have will be working with probes that are just that sensitive. So we saw that you don't even need gel electrophoresis for this. You simply see where probe number 1 sticks. Those are your homozygous, heterozygous, and homozygous mutant individuals. Boil the nylon piece of paper. These days, you'll see there's a chip, a glass chip to which these DNAs are affixed. Boil the probe off and then probe it again with the other probe. This kind of technology that you can use over and over and over again. The hospitals own thousands of these chips that have little stretches of DNA covalently bonded to them, and you just change the probe that you slide over these day after day. This kind of technology, which they call digitized, the probe either sticks or it doesn't. So yes, no sort of technology. And we don't need the gel electrophoresis, which reduces the time and the expense in a lot of cases. So this was an example of that. Then we moved on to a new topic, entering the main section of the course where we talk about replication, transcription, translation. So we started off with replication, and we defined terms semi-conservative, bidirectional, symmetric, semi-discontinuous. Students are sometimes on an exam asked to explain what these words mean. Often they are asked at the same time as being asked, and what word would you use for RNA transcription instead? And we haven't talked about that yet. I think we won't get to it till Wednesday. But I just wanted you to be aware this happens a lot again in college. We teach you something on one day, we teach you something else on the day a week later, and then on the test we ask you to put it all together maybe for the first time. So yes, at college we do try to see if you can integrate topics and not just repeat back what we said to you. So this will be something that we will contrast with replication. That'll get you in the habit for contrasting replication and transcription, contrasting that with translation. So we thought that really the main purpose of the entire course is to not mix these three processes up. I just think this movie is remarkable. I can draw it on the board, and it's not as realistic as this cartoon can be. So in this cartoon, and you could certainly look this up online, there's a narrator and everything, but there's the helicase, there's the sliding clamp, there's the DNA polymerase for extension, there's the primer you can see, leading strand, lagging strand. There's a lot that you can label on this kind of a living diagram, which is an awful lot of fun to see. They say it's real time, at least as far as they can tell. How is DNA replicated? That lecture we had last week where we talked about semi-conservative, bidirectional, asymmetric, semi-discontinuous, and we mentioned all the enzymes that participate in this process. That's a big part of your undergraduate curriculum in molecular biology. In fact, this question is on the exit exam, so seniors before they graduate have to take a little test, and they're asked, how is DNA replicated? We assume everyone heard about it in 201. You hear about it again in genetics, again in genomics, again in cell, again in biochemistry, so how is DNA replicated? But we're sort of expecting an essay, kind of like this. And notice the words in bold, these are the words that came up. It's not, the book will say, how is DNA replicated? They'll have 50 pages in a chapter. This is the kind of thing we would expect students to say. You bring up semi-conservative, complementary, primase, primer, DNA polymerase 3, DNA polymerase 1, Okazaki, leading, lagging, RNase H. All of it's there. So that's a pretty high quality answer without taking up a book. I don't think you could copy this out of a book. You kind of have to show that you understand these things, know that they're important, and can put them in the right context. But that's the kind of thing we expect a senior to be able to do. So maybe this is the second time you've heard about replication. Maybe you'll hear about it once or twice more. But this is one of four main things we expect majors to understand. Snap a picture of it and get ready to graduate. DNA polymerases. We've got Pol III, we've got Pol I in a prokaryotic system, and we already know pretty precisely what they do. We said there's 20 different DNA polymerases in eukaryotic systems. I don't think anyone really has written down a list of all 20. I presume they specialize. That's what we tend to find in eukaryotes, many more proteins, each doing a smaller part of the job. But DNA polymerases in general, and again, okay, maybe you can hear it without me saying it, but I'm going to say it out loud so you know. If we're talking about DNA polymerases today, the purpose is so that next time when we talk about RNA polymerases, you're already saying they're not the same and here's the ways that they're different. Okay, so this is meant to be on 2 different days, but you're meant to integrate this together, so just be prepared. DNA polymerases, they require a primer. You have to have a template. You have to have the enzyme, you have to have the DNTPs, the monomers. But even if you have all that required stuff and put it in a test tube, nothing happens. You have to have that primer offering the correctly paired 3' hydroxyl. We drew it in green last time. RNA polymerases aren't going to need no primer. They're just going to do their job. They could care less what happened a second ago. Whereas the primer requirement means that DNA polymerase is always looking, did something happen correctly right back here? Okay, then I'll go forward. So it seems like, well, why is RNA polymerase so smart that it doesn't need a primer, but DNA polymerase is so dumb that it needs a primer? We've already talked about one RNA polymerase. Primase is itself an RNA polymerase, and it just got started. Single-stranded template, ribonucleotides in the vicinity, boom, it starts to lay down a primer. The problem is that RNA polymerases offer low fidelity, DNA polymerases offer high fidelity. So all DNA polymerases have the primer requirement. They can only extend an existing 3' hydroxyl that's already paired to a template. They all will polymerize only in one direction, only in the 3' direction. That makes sense because they're looking for that 3' hydroxyl and then they go on. Then they're looking for the 3' hydroxyl and then they go on. A required part of that condensation reaction that we talked about. This might be a little surprising. All DNA polymerases can turn around and go back the other way and cut out the thing they just added. And maybe that's not too surprising because that's how biochemistry works, right? Substrates go to products, but you can take the products and tear them down and make the substrates. Biochemistry can run in either direction. So maybe it's not surprising that an enzyme that can polymerize in this direction can be an exonuclease in the other direction. It's really the same process. But these 3 characteristics of all DNA polymerases add up to the high fidelity, which maybe you guys don't even know what that word means. Back in the days of vinyl record albums, that they all advertised that they were high fidelity. It almost sounded exactly like The Who playing live, but it really didn't. So high fidelity means DNA is making a very faithful replica of the template. With only 1 in a million mutations is sort of the number that we imagine. And so these 3 requirements of DNA polymerases really leads to the idea that DNA replication should be high fidelity, very few mistakes. So that what the enzyme, I'll be the enzyme, the floor will be my template, I'll be wanting to polymerize in this direction. It literally is, it's looking behind for a 3' hydroxyl in the previous base or on the primer, which is in the right location, if there was a purine across from a purine, the hydroxyl would be way too high, right? Because purines are big. Well, DNA polymerase can't go looking for it. If it's not there, it turns into an exonuclease and throws that out. Oh, now I find the hydroxyl where it belongs, and now I can put one in, check, put one in, check, put one in. Oh, until I put a pyrimidine across from a pyrimidine. They're small. Now it's too low. I'm not allowed to go fishing for it. If it's not there, turn around and throw the last thing out until I find things in the right place, and then I can go forward again. So the presumption is things like primase can put the right thing in, can put the wrong thing in, doesn't really matter. There can be no primer there, and it's gonna lay down a complement and attach it to whatever it can. So it sounds like, wow, it has this great ability to not require a primer, but it's gonna do sloppy work therefore. It's not checking to see that the 3' hydroxyl is precisely where it belongs. And so these characteristics of all DNA polymerases is why DNA polymerases are high fidelity. And again, we'll see why this is sensible. DNA that we synthesize basically lasts forever. RNA that we synthesize is around for 2 minutes at the most. So if you make mistakes, you're gonna make another one 2 minutes later. Whereas DNA polymerase, you make a mistake, you and all your ancestors have to live with that change or die from that change. So DNA polymerases make a high fidelity copy. In addition, DNA polymerases, to different degrees, also have a 5' to 3' exonuclease activity, which we described for DNA polymerase 1. If it detects in front of it damage, it can get rid of that damage while it lays down good DNA behind it. So for example, that RNA-DNA duplex left behind by the previous primer, DNA polymerase 1 will say, that's wrong, you don't want RNA and DNA, and it'll clip that out in the front, going in a 3' direction as it lays down good bases, again, going in a 3' direction. Now, for eukaryotic polymerases, I kind of expect they're all gonna have these characteristics, but we may have specialists. Some of those 20 DNA polymerases in eukaryotes may show up at precise moments during the cell cycle or under precise environmental conditions. They probably do divide up the job in some particular way that I haven't read about in great detail. So for our purposes, when we are talking about eukaryotes, we'll simply say A, DNA polymerase for repair, A, DNA polymerase for extension, and that's enough of a specificity for our purposes. Okay. Something new for today to get us ready for tomorrow. And I guess we'll see how far through this I can get. And if I don't get all the way through, we can pick this up at the beginning of lab tomorrow as well. One of the things we'll do tomorrow is the polymerase chain reaction. It is it's a laboratory technique that is basically DNA replication in a test tube. And so it's a good time to teach the polymerase chain reaction so we can see the things that a cell does, how we can exploit those same steps and some of those same enzymes in a test tube environment so that we can carry out certain experiments. Let's see, let's think of the ingredients. One of the things you need is some template DNA. Now, the thing about the polymerase chain reaction is everybody does it a little bit different. I think every biologist in the world is doing PCR for different purposes. What we teach is sort of the standard approach to PCR. So the template DNA in a standard reaction is frequently human genomic DNA. So it's a good place to start. Remember the Southern blot, we had to find in that whole human genome the one thing we were looking for. The polymerase chain reaction has the same purpose. So let's imagine we're starting with human genomic DNA in our test tube. And not very much of it. Let's have a very small amount. Sometimes you'll see on court TV, the lawyer allows the opposing team to get like 3 hairs off of the suspect's head.
get the DNA out of the roots of those 3 hairs. So there's not much DNA to work with. The other thing we'll throw in, I guess I can draw that. So there's my template DNA. Double-stranded. And I'm only drawing a little of it. I suppose in reality, if I was depicting the genome, it would go on for miles and miles and miles and miles. We're just looking at the part that we care about for this purpose. Template DNA. The part I care about is going to be defined by the primers that I use. One of them is said to be a forward primer. One is said to be a reverse primer. These primers, it's similar to the primer we just talked about in replication, except these are single-stranded DNA molecules that are synthetic. Synthetic, which means some biochemist in a lab made them for you. Synthetic single-stranded DNA. They're not really polymers. We call them oligomers. They tend to be about 20 nucleotides long, which is not really poly, it's just a few. So synthetic single-stranded DNA oligomers, they're going to serve the same purpose. They are going to prime replication in a test tube. But they are not synthesized by primase in this case. They are not made out of RNA in this case. Let's see. One of these primers is going to be precisely the same as this right there. One of these primers is going to be precisely the same as that right there. And so if I know this DNA sequence, I can call up my biochemist supplier and say, send me a forward primer. I want these 20 bases, and I'll rattle them off 5 to 3. And then in a second test tube, send me 20 single-stranded DNA bases, which are in this order, and I'll rattle them off 5 to 3 that way. So they're in my test tube together with my template DNA. Now, if I'm going to carry out replication, I certainly need a DNA polymerase. If I'm going to synthesize DNA, I certainly need some deoxyribonucleotide triphosphates. Those are my monomers. My ATP, GTP, CTP, TTP, those are my monomers. The other thing we're going to go through is the conditions for this polymerase chain reaction. The first thing we'll do is we will go to 95 degrees Celsius, and this is called the denaturation step. And so I can depict what happens now that all my ingredients are in my test tube. I'm just drawing one of them, and I'm just drawing it up close in this little single area so we can see what's happening. If I go to 95 degrees, I will simply separate. And this may be hard for you guys to draw because I'm going to erase and move things from time to time. It's going to be hard for me too because nothing erases up here. Okay, so I'm trying to draw my template DNA in a slightly different location than last time without changing anything about it. In the next step, so maybe that happens for 30 seconds. In the next step, we're going to go to maybe some cooler temperature. I'm going to make one up, 55 degrees. This is sometimes called the annealing step. It's going to last 30 seconds as well. So we're going to go to a nice cool temperature. First we've denatured our templates, now we're going to go to a cooler temperature. Now, this is cool enough to allow DNA to snap back together, but this is the whole genome. It's very complicated, and I implied that it's very dilute in my test tube because I only got one hair from Michael Jackson's head. So what is abundant in my test tube are these forward and reverse single-stranded DNA oligomers. They are going to lay down on their complementary anti-parallel target almost instantly, probably in a millisecond or a few milliseconds. Whereas the template strands that I've put asunder are likely to require weeks or months to find themselves. And I'm only allowing a few seconds. So, most common question for students is where does the red primer go? So So the forward primer is red, the reverse primer is blue. Where do they go? And almost everybody points up here and says this is where the red primer goes, and that's wrong. Where does the red primer go? The red primer is exactly the same as that, therefore it's going to stick down here where it finds an anti-parallel complement. If my blue primer is exactly the same as that, it's not going to stick there. It's going to stick to its anti-parallel complement. And almost everybody gets that wrong. And that's probably the most commonly asked question. Once the primers have all laid down, then we're going to go to our third temperature. This might be 72 degrees C or something else, but it's warmer. This is called the extension temperature. And maybe that's only going to last for 30 seconds as well. That happens to be the temperature of the water in the geysers at Yellowstone National Park. Because they found some bacteria living in there that love 72 degrees, and they don't even mind going to 95 for a brief time or 55. They really love 72. And so that's the bacterium from which we have isolated the DNA polymerase. Frequently this goes by a name. This is the DNA polymerase from Thermophilus aquaticus. So the shorthand is TAC. Thermophilus aquaticus is the name of that bacterium. So TAC DNA polymerase is what most people will use. If we use DNA polymerase from E. coli, which likes what temperature? 37 degrees, because it lives inside of me. Every time I go up to 95 degrees, I'm going to have to start over and put some fresh enzyme in there because I denatured my enzyme. So the beauty of the TAC DNA polymerase is it doesn't mind these high temperatures. And so what will happen at the extension temperature, the TAC DNA polymerase will find that 3' hydroxyl, will find DNTPs in the vicinity, and it will extend these. And it won't go to the end of the chromosome because I'm immediately going back up to 95 degrees. And that's going to denature everything. So I will draw these new molecules in a slightly different location just to represent denaturation in my second cycle. So synthesized lying on top of the template, but then melted so that they're someplace asunder at this point. Then immediately we go to 55 degrees, and my primers will stick. My primers will stick here, my primers will stick here. That's twice as many places that I had to draw a red primer. My blue primers will stick here and here. That's twice as many places as I had to draw blue primers last time. So we're trying to build this idea that this is a geometric progression. We're doubling. At 72 degrees, TAC DNA polymerase will extend, just like we did last time. But now we're going to extend on this shorter template, and we're doing that for the first time. And then we're right back up to 95 degrees, and so I have to move everything again. Let's see if I can do that. This will be the last time, and we'll go to a previously drawn picture after that. So everything denatured. Then we're back at the annealing temperature. Now we're going to stick red primers again. There's one. There's two. There's three.
I think you're missing a line at the
Yeah,
top.
I'm doing something wrong. I don't like that these are all facing the same way. Yeah, there should be four. I'm not going to figure out what I did wrong now. Let's see. But my blue ones, again, are going to go here, and here, and here. Twice as many places. So the idea is the annealing is going to be just 2, then 4, and then 8. So we're doubling the number of spots to which the primers anneal. Doubling. And we're also supposed to notice that when I extend these, even in this round, we're going to go a little bit past and make these somewhat larger molecules only twice. We made 2 in the first round, 2 in the second round, 2 in the third round. You can imagine we're going to make 2 in every round. Whereas these precisely sized ones, like that one, like that one, like that one, like that one, we make more and more of the precisely sized ones to the point where we're making 2, 4, 6, or 2, 4, 8, 16, 32, 64. We get very quickly, after 30 cycles, 1 billion molecules that are exactly the space between the red and blue primers. And each cycle, so 30 cycles, we're making 2 more molecules that are a little bit bigger. So after 30 cycles, we have 60 molecules that are, I don't know how big, bigger than my target region. But I've got a billion that are precisely the size of my target region. Now, on an electrophoresis gel, can I see 60 molecules? No. On an electrophoresis gel, can I see a billion molecules that are all the same size? Yes, it's going to give me a nice bright band. And so these are the details of how the polymerase chain reaction actually goes. Sometimes students are asked about the ingredients. Sometimes they're asked about what happens at these conditions. Drawing this is a mess. And so we'll look at something from the book instead, but we'll see, I think, all the same hallmarks. Yeah.
What's stopping the Taq polymerase from going past the region that you all specified?
On these, my template strand is so short that it runs out of instructions. On these ones where the template is very, very long, well, nothing does stop it except I suddenly denature the whole thing. It has no choice but to quit when I go to 95 degrees. I think we can see all those things. So the region to be amplified is in light gray, and they're showing a tiny little bit of DNA on the outside. It's literally miles. Step 1, denature. Step 2, anneal, and you can see the forward and reverse primers are defining the region of interest. Step 3, DNA polymerase will extend, and it goes past the region of interest, but only those 2 molecules go past. Then we pick it up where we left off. We denature. We got a couple of these sort of long molecules. We're annealing in twice as many positions. Now we are making a precise length. For the first time, just 2. And we're making the kind of long ones again, but just 2. We denature again, and now they've highlighted all the ones that are of a precise length. And we can find there's just a couple that are longer past the region of interest. But if we are doing an arithmetic projection by having 2 plus 2 plus 2 plus 2 after 30 rounds makes 60, if we're doubling 30 times, 2 times 2 is 4, times 2 is 8, times 2 is 16, we get to 1 billion in 30 rounds. So what people typically do is 95, 55, 72, and we go back and call this another cycle and another cycle. We go through these cycles about 30 times to get to that about 1 billion copies. Now on this diagram, they are counting these things up for you to kind of prove